talked a good bit about membranes, and it just occurs to me I've not prayed. Let's pray. You all would join me. Heavenly Father, thank you for this morning. Thank you for these who are able to be here with us, and those who will watch later, watch this video. Would you please, by your grace and by your Spirit, help us to learn these things, these processes that happen in our cells, the stuff moving back and forth. We can't live without this. This is literally the stuff of life, and you put it together. Each and every protein, carrier, transport, lipid, you designed it all to work. It works just so amazingly well. You're sovereign over it all. May we catch something of the awe of you in the intricacy and the beauty of these little tiny molecules that keep us alive. We pray for your grace for our soccer players and our golfers, Lord. Keep them safe on their travels. Let them have good success in their endeavors and also with their academics. Lord, they'd have a good time. Keep them from illness. Keep them from harm. We pray these things in Jesus' name. Amen. We've been talking about cells and membranes now for a little bit. Of course, a cell is not a cell if it doesn't have a membrane. And one of the things about a cell is it's got to keep you alive. It has to maintain homeostasis. We've talked about this word a good bit. Homeostasis. It's the idea of maintaining consistent levels, consistent environment. If you take in food, right? You take what you need out of it, then you always have waste products, which you have to get rid of. If you can't get rid of waste products, you die, right? I have diverticulitis. Young for diverticulitis. Diverticulitis will kill you. You get a little infection in your colon. And if stuff can't pass out, everything swells up and your gut explodes and you die within an hour. You must be able to get the good stuff that you want in and the bad stuff you want out. That's at an organismal level, but it's also at a cellular level. If the cell can't get rid of just the junk, the metabolic waste, it will die. If I took a saltwater fish and I threw it into a lake, what's going to happen to it? Say die. Say die. Yeah. It will die, right? Because it needs salt water. It's adapted for salt water. Conversely, if I take a freshwater fish and I throw it in the ocean, it will die because it does not have what it takes to pump all that salt back out, right? It has to be able to maintain homeostasis. I'm sure I told you earlier in the year that more people die in the world every day of dehydration than of anything else. Actually, of everything else combined because of homeostasis. They can't maintain the right balance of water inside when it's all being lost, whether it's through their bowels or whether it's through sweating. It becomes a problem. You have to maintain homeostasis. So how does this fit? Well, if I'm going to maintain homeostasis and keep the inside things in, or get the inside things in and get the outside things out and keep them out, I can't just seal myself into a room like this with no way in or out, right? Stuff has to be able to get in and out. And that's where the cell membrane comes in, right? And so we've talked about this. Some things move very freely in or out of a membrane. They simply move down their concentration gradient. So if I've got a bunch of red dots out here, and very few red dots in here, which way do red dots want to move? In or out of the cell? Well, I've got a lot out and very few in. They go down their concentration gradient, so they want to flow into the cell, right? Conversely, compared to red dots, I've got a lot of water inside the cell and very little outside the cell. Which way does water want to flow, in or out of the cell? It wants to flow out, down its concentration gradient. So now think about this. If I'm a fish, and this is salt, and so here's my cell, and this fish that just got thrown into salt water, and I'm about to die. I'm about to die because the salt is going to flow in, my water is going to flow out, my cell is going to shrivel, I can't keep up, I'm in trouble, right? It'd be really good if we had some mechanism for pumping salts in and out, because salt left to itself is just going to flow down its concentration gradient. Hopefully you remember this, especially with somebody watching. When something moves down its concentration gradient, what's that called? Starts with a D?
Dehydration.
No.
Denatured?
No. Diffusion. Diffusion is when something moves down its concentration gradient. So it's part of what we call transport, right? So transport being the movement of any molecules across a membrane. We've got two types of cellular transport. Maybe I should write that up. There's cellular transport. Cellular transport. So one is when it just flows, right? That requires no energy. This is passive transport. And the other is maybe I believe would like to flow down its concentration gradient, but there's no way for it to. Maybe the cell is just not very permeable to it, or more often I need to move it against its concentration gradient. Okay? Imagine being at a sand dune. Okay, which way? And here's you down here. You're smiling, you're happy. And your job is to get rid of the sand. But what does the sand keep doing? At a sand dune, right here. What does it keep doing? It just keeps flowing down to you, right? And you keep you've got a shovel. This is you with your shovel working very hard. And you keep shoveling it up. But it keeps falling down. You keep shoveling it up. And it keeps falling down. You keep shoveling it up. This is like a saltwater fish in the ocean. The salt is constantly flowing in. It's got to get the salt out or it too will die. It's just better at it than a freshwater fish is. And so it's constantly, it's like swimming upstream. You're shoveling the sand out in this case and it keeps coming back in. In this case, you're pumping the salt out against its concentration gradient, but it just keeps coming in. But if you ever stop, you die. You've got to fight this. Well, that is not passive transport, that is active transport. Active transport requires energy. We've talked about this just a little bit. We've talked about the energy source for this being ATP. And right now that's just 3 letters to you. I'm going to show you a video, Lord willing, later on, depending on the pace of class. And we'll show you what ATP looks like and kind of how it functions and contributes to all this. So this requires no energy. This requires energy. Passive transport, there's simple diffusion. Okay? There's osmosis. And then another would be, maybe we call it gated diffusion. So diffusion, of course, is simply something moving across its concentration gradient from high to low concentration. Some things can do that in a cell very well, very easily. Some things cannot. I'll give you a second to catch up with me, okay? So, to understand which things will and will not make it across a cell membrane easily, I need to draw another picture of a cell membrane. Do you remember the thing that it's made up of most? If I had to draw this, what do I need to have a lot of? Phospholipids. Good. So, phospholipids are these, remember we got these phosphate heads. What's the charge on these? Do you remember? Are they neutral? Do they have a charge? Positive, negative? Are they polar or non-polar? No.
Polar.
This part's polar. So these are actually have a slight negative charge to them, okay? All right. And then I've got these tails. And what are they? Polar or non-polar? Are they hydrophilic or hydrophobic? These are hydrophobic, right? Water theory, no charge. Let me draw a few more of these, okay? Now, We talk about the cell all the time. It's called a phospholipid bilayer. 2 layers of it. So when you look at a cross section of a cell membrane, is most of this hydrophilic or hydrophobic? Does more of this have charge or more of this not have charge? Yeah, most of this is hydrophobic. Only the albumin is not going to make it across this very easily. To make it across the hydrophobic region, I need something that's hydrophobic. I need something that's not charged, something that's electrically neutral. So something like oxygen has no charge. Oxygen gas will easily diffuse across this nonpolar membrane. Okay? Something like carbon dioxide. Here's your cell. You got, usually because of your breathing, you've got oxygen in, you got carbon dioxide out. Oxygen will readily diffuse in, carbon dioxide will readily diffuse out. It just moves down its concentration gradient because it's not charged. And it's also very small. That's another. So things that are going to move by diffusion are going to be small and nonpolar. If it's big, it's not going to make it. Glucose is nonpolar, but it's big, so it needs help getting across. Sodium and potassium and hydronium ions, they're very small, but they're electrically charged, so they are going to need help. So that's where this idea of like gated diffusion comes in. And so things that happen here are things like water. Potentially something like glucose. Okay. Now I've got water here, but of course water also goes here under osmosis, right? So do you remember a decent definition of osmosis? It's the movement of water across a selectively permeable membrane. This is a definition you must know by October 1st, right? You must know it. You just count on that. That will be on a test. Movement of water across a selectively permeable membrane. When you have that written, put your pencil down because I just remembered I need to remind you of something. So you do have a test coming up. We really, really need to do well here, okay? Oh, this is October 1st. You have a massive homework quiz up, okay? Just went up a day or two ago. You can take as many times as you want. Remember, this is not, I'm going to give you get the grade you want, right? Take as many times as you want. The big part is not the homework component of it. The big part is how it's going to help you for the test, okay? It is set up all the same information that your test has. Okay? It's going to ask you things like, is this organelle membrane bound or not? Is this organelle found in a prokaryote, a eukaryote, a plant, an animal? Those,
just the organelles.
On the homework or on the on the Moodle? Good. Did it go okay? Okay. Well, that's all right. So that's okay. You want 100. Right? So, and one thing is there's a page in the textbook that has like a chart of all this. I didn't even go off of my own, though I've given you some stuff like that. I literally went off the chart in your textbook so that you can just pair it. Like I used the exact definitions that they used to just keep it simple and consistent. So if you go study that chart, that's really going to help. Okay? And again, you don't.
So that's not for this test I'm about to take after classes.
Are you taking it today?
That was.
I didn't know. Okay. No, it will not be on that one. It will be on the next one.
Okay.
Okay. All right. So do that, okay? Go take that and get to where you can kick its tail. All right. So we have active and passive transport. When we talk about this, we're talking about just how do we move stuff in and out of the cell. Passive, no energy. Active requires energy. Types of passive: diffusion simply moves down its concentration gradient. Osmosis is the movement of water along its concentration. It's just diffusion, okay? And then there's gated diffusion, which is still diffusion, but of course, in gated diffusion, it's got to move through a protein, okay? Through some kind of a channel or a gate, a protein. That's the idea of this. When we say gated, it's gated by a protein, okay? And then there's active transport, and as it implies, it requires energy. And the reason it requires energy is because we're going against the electrochemical gradient. Moving something against the electrochemical gradient. There are many, many instances in your body that you want to do this, you need to do this, you have to do this. Fortunately, your body is really, really good at doing this. Oh my goodness, my nose. Okay. Let me just make sure before we go to these videos that I'm not missing anything I need you to know. Good description of a cell membrane, a model of a cell membrane. It's a word you've heard from me.
You said a model?
Pardon?
You said a model?
A model of a cell membrane? Yeah. With all these parts that move around, kind of looks like a picture. The idea of movement, things that flow or move, what do we call those? Fluids. And it's like a picture made up of a bunch of little parts. We call it, they're on like the floors in Rome and Greece and stuff. We call those mosaics. It's all good. This is for the next test, right? So, fluid mosaic model of a cell. It's the idea that the parts move, and we've got a mosaic of things. That's even going to come up in the video in a minute, so that'll be good. So, fluid mosaic model, and of course the parts of a membrane, to review, phospholipid bilayer is number one. That's the background to everything. And then I've got these proteins, and I've got these carbohydrates, and maybe the one other major thing that we'd want to talk about is cholesterol. It's hydrophobic, so cholesterol, and they're going to show you cholesterol even in this video, which is really good. So it's this is something you need to know for your test. It's 4 fused carbon rings. What's a lipid with 4 fused carbon rings called? Huh? Steroid. There you go. That's it. So, and look, this will help too because you're going to have a picture of it, right? So, embedded in this phospholipid bilayer are lots of, are potentially a lot of these. Oh my goodness. I'm not good. Lots of these steroids, which are called cholesterol, right? And so the thing with these is they slow down how fast this moves. Or you take them away and it speeds it up, right? Okay. Let's see, what else do we need to look at before we go to that? Diffusion. This is also called facilitated diffusion. I guess we should know that. I'm not going to get hung up on all the. Did you use the exact word that I was? I won't do that to you. Okay. Okay. Osmosis. Oh, we do need to review hyper and hypotonic and isotonic. Okay. So related to the concept of diffusion. I've got a selectively permeable membrane here, so some stuff can move across it and other things cannot. Right? And I've got dots, because I like drawing dots. The dots are my solutes, water is my solvent. I have lots of dots on this side, I have no dots on that side. This side is called what tonic? Hypertonic. I have a greater concentration of solutes as compared to the other side. And this side is hypotonic. And diffusion says that the solutes want to move from what side to what side? Hyper to hypo until they are isotonic, right? So let me draw one more here. We'll do isotonic. So this is going to be isotonic. So where they're on the same, right? But so often osmosis is also happening, and so water wants to flow from one side to the other. Well, if my solutes can't get across, but water can, what's going to happen to this side? What's all the water going to do from over here? It's going to go to the other side. Water wants to flow over here, and it will do that until they are also isotonic. This matters, because if I'm looking at a cell, and the cell is not permeable to red dots, but it is permeable to water, then what's this cell going to look like after about an hour? No? If it's not permeable to red dots, what's the water going to do? Water's going to flow out. So what will the cell do? It'll shrivel. Denaturing is only the changing of shape of the protein. Okay, that's when it loses its shape or its conformation. So this cell will shrivel. The opposite is also true. Oh, by the way, in this environment, since I'm losing water like crazy, what do I have to be able to do? I got to be able to pump water back in, right? Through active transport, otherwise I'm going to die. So I can fight what's happening if I apply energy, right? Remember the very first class, and I made you guys like repeat after me as this weird chant, like, I am an anti-entropy machine. Well, entropy is what's causing this. Everything flows from high to low concentration, but you're so good at fighting entropy. You're like an anti-entropy machine. You can pump the water back in. If you ever lose that ability, you die, right? Okay, so in this case it shrivels. But there's another option. What if I have this? I've got all these red dots in here, and I've got no red dots out there. I'm not permeable to red dots, nor do I even care to be. What does water want to do? Flow in or out? It wants to flow in. What's going to happen to this cell after an hour?
It's going to be huge.
Well, it's going to be beyond huge. It's going to pop. It's like a balloon. If I just keep blowing into it, right? So we'll just have like fragments everywhere, right? It'll do what's called lysis.
Did you say lysis?
Yeah, split. Hydrolysis is to split the water at the chemical level. Okay, so this will lysis. So a cell has to be, this cell would have to be able to pump water out. This is kind of like what's happening with a freshwater fish, right? So saltwater fish has to be able to pump water in and pump salts out. Freshwater fish has to be able to pump salts in and pump water out. They've got to go against the concentration gradient. This is the whole idea of anti-entropy machines, of maintaining homeostasis. It's a form of active transport because you've got to fight the passive transport. Okay? All right. Well, let's see what else we got for this. Perfect. This is where we are. This is where I want to be. How long did that take? We did that in half an hour. That's great. Okay, I'm going to show you guys some videos. Okay, these videos are going to demonstrate particularly, well, one of them is going to be an overview of everything, and then the rest of them are going to kind of look at active transport. And of course, we'll stop and we'll talk about them and we'll review them. These are up on the Moodle, so you can go back and look at them later. I'm telling you right now, you must memorize and know what's happening with the sodium-potassium pump. Just write that down. I must know and understand the sodium-potassium pump. It's going to be kind of our model for active transport, okay? So, let me,
I must feel what?
the sodium potassium pump. So, let's do this. You okay?
I had paper last night and I didn't turn anything
Oh.
I don't know who's looking
Here we go. Overview of cellular transport animation. Okay.
Part of metabolic activity is the ability to move materials into and out of cells. This is important in cell communication and normal cell function. For example, in order for the cells of your nervous system to function properly, ions, water, proteins, and other molecules need to be able to pass into and out of cells. The cells of the nervous system form networks. They, like all cells, are able to function because they can control what substances are inside the cell and what substances stay outside the cell. These materials move in and out of cells by passing through the plasma membrane of the cell. The plasma membrane surrounds the cell and separates the interior of the cell from its environment. This semi-permeable membrane is composed of a lipid bilayer. Phospholipids make up a large part of the membrane and form a bilayer. The structure of the bilayer is due to the tail-to-tail packing of the nonpolar hydrophobic tails, composed of 2 fatty acid chains, and the polar hydrophilic heads, composed of glycerol and phosphorylated alcohol. The lipid bilayer is 5 to 10 nanometers thick and is embedded with proteins. Some cell membranes also contain cholesterol. A plasma membrane contains different types of proteins which are specific to the particular function of the cell. These proteins also enable the cell to interact with its environment. The entire structure of the plasma membrane can be described as a fluid mosaic model. The phospholipid bilayer has properties resembling fluids, and the differing proteins and their attachments on either side of the membrane resemble a mosaic. The plasma membrane is the gatekeeper of the cell, allowing certain substances in and out of the cell at certain times in certain amounts. Diffusion is a process in which substances move across a membrane from an area of high concentration to an area of lower concentration, or between areas of opposite electrical charges. This is called the electrochemical gradient. Small, non-charged particles, primarily gases such as oxygen and carbon dioxide, can diffuse through the plasma membrane by moving in between the phospholipids in the bilayer. However, the cell needs to control what enters and leaves, and so transport proteins aid in the selective movement of other molecules across the membrane without the input of energy. Through facilitated diffusion, larger molecules, polar molecules, and charged ions.
So they use the word facilitated diffusion, right? So first thing we want to think about there is the word diffusion. So that just means it's going from high to low, right? You can see here on this video, lots of potassium, purple, inside the cell, relatively little outside the cell. So potassium wants to move down its concentration gradient as much as it can. Problem is potassium is charged, okay? And that cell membrane, the interior of it is not charged, so it can't just flow across. It has to have something to help it, something to facilitate it, hence the term facilitated diffusion. And that's where this protein channel works, right? So it's not passive, or I mean it is passive, but it's through a protein, but there's no energy involved here. The protein just simply makes a pathway for it. So hence facilitated means to help facilitated diffusion. That's what's going on there.
Use channel proteins embedded in the bilayer. The transport of other substances requires a special carrier that will bind the substance on one side of the membrane, which triggers a conformation change in the protein carrier, causing it to release the substance on the other side. When the substance being moved across the membrane is water, the process is called osmosis. The cytoplasm of the cell, as well as the interstitial fluid, is composed of solutions. The solvent, usually water, moves across a semi-permeable membrane toward a higher solute concentration, consisting of various molecules or ions, until equilibrium of the solutions is reached. The plasma membrane contains proteins called aquaporins, which are specialized channels for the movement of water during osmosis. A cell in a hypertonic environment will have water move from the inside of the cell toward the higher concentration of solutes in the solution outside the cell. In a hypotonic solution, the concentration of solutes is higher inside the cell than the outside environment, so water will diffuse into the cell. When the solutions on either side of the membrane reach equilibrium, they are referred to as isotonic. When substances need to be driven against their concentration gradient from low to high concentration,
What kind of transport is this going to be? It's going to go against its concentration gradient. We're going to go from low to high. That's going to require ATP. It's what kind of transport?
Active.
Active. Good. And actually, this is a little diagram of, we're going to see a better video than this, but this is the sodium-potassium pump they're showing you.
concentrations, active transport is required. This process uses energy to move a substance across the membrane using selective protein carriers. The sodium-potassium pump is a specialized membrane protein carrier, common in most animal cells, that moves sodium and potassium ions across the plasma membrane. ATP fuels the pump in the movement of these ions from low to high concentrations, moving sodium ions outside of the cell where they become concentrated and bringing potassium ions into the cell where their concentration is higher relative to the outside. In other instances of active transport, the concentration gradient created from the action of one pump can be used as the input of energy to drive a different pump, a process known as coupled transport. The large concentration gradient of sodium ions is used to bring glucose into the cell against its concentration gradient. The plasma membrane, with its lipid bilayer and various embedded proteins, provides a barrier between the internal cellular environment and the environment surrounding the cell. The structure and function of the plasma membrane, coupled with the process of moving materials into and out of cells, as well as for the continuation of life.
Questions about anything you saw right there? Everything's good? If it requires ATP, what kind of transport? Active. If it does not, it is passive. Okay. If it requires a protein, a special protein to get through, it's called still passive. Well, what kind? Facilitated. Means help, facilitated diffusion. If it can just do it on its own, it's just regular old diffusion, right? Diffusion of water across a selectively permeable membrane is called osmosis. Let me answer those questions again. By the way, when you hear me ask questions like this, you're hearing how I'm thinking, right? This is how I think about the thing, this is how you need to think about the thing, because this is how the test will think about the thing. If it does not require energy, what kind of transport? Active or passive? Passive. If it does, it's called active. Referring to the passive, if it needs some kind of a protein carrier or channel to get across, what's that called? What kind of diffusion is that? Facilitated diffusion. If it's the movement of water across a selectively permeable membrane, it's called osmosis. If it's just some small nonpolar molecule moving across down its concentration gradient, it's called just diffusion, right? What kind of things cannot, even if they're going with their concentration gradient, they can't just get across the membrane? it's got a charge to it or if it is big. Yeah. Glucose can't do it because it's too big. And salts, sodium, potassium, these things can't do it because of their charge. Right? Water can't normally do it because of its charge. Water, though, has these, they talked about here, aquaporins. It's like a gate, a channel that water can just flow through. Which particular active transport protein are you about to learn about that you must learn? I mean, you gotta just know it. Sodium, potassium, pump. Okay, so we're gonna learn about that in a minute. Again, all of this is up on the Moodle.
Do you know what's really fun? Check your website on Wix.
The sodium-potassium pump is an active transport mechanism that is driven by the breakdown of ATP and works through a series of conformational changes in the transmembrane protein. 3 sodium ions bind to the cytoplasmic side of the protein, causing the protein to change its conformation. In its new conformation, the molecule becomes phosphorylated at the expense of a molecule of ATP. The phosphorylation induces a second conformational change that translocates the 3 sodium ions across the membrane. In this new conformation, the protein has a low affinity for sodium ions, and the 3 bound sodium ions dissociate from the protein and diffuse into the extracellular fluid. The new conformation has a high affinity for potassium ions, 2 of which bind to the extracellular side of the protein. The bound phosphate now dissociates, and the protein reverts to its original conformation, exposing the 2 potassium ions to the cytoplasm on the inside of the cell. This conformation has a low affinity for potassium ions, so the 2 bound potassium ions dissociate from the protein and diffuse into the interior of the cell.
Just chilling with Frontier Fiber Internet and YouTube TV. I'm not.
The sodium-potassium pump works constantly to maintain a low concentration of sodium ions and a high concentration of potassium ions in the cytosol. This is achieved through the following steps. The sodium-potassium pump binds 3 sodium ions from within the cytosol. ATP then binds to the ATPase region of the pump and is hydrolyzed to ADP and a phosphate. This phosphate remains bound to the pump, phosphorylating it. Phosphorylation causes the pump to change in shape. The sodium ions are released into the extracellular fluid and 2 potassium ions bind to the pump. The phosphate group is then freed from the pump, causing the pump to return to its original shape and release the potassium ions into the cytosol. The pump thereby returns to its initial state, ready to repeat the process.
Y'all ready to draw?
Is that bothering you? Okay, it just seemed loud. Okay.
Thank you. Okay, guys. So, we've got a cell membrane. Somewhere in here, I've got this pump. That was terrible. cross section, right? That's the idea here. This marker's showing. Okay. So I've got this pump. Now, the assumption is I've got sodium on the inside. Okay, sodium is going to be squares. And I've got potassium inside. Notice I've got a lot of potassium inside compared to outside. Okay. And I have relatively few sodium inside compared to outside. Which way does sodium want to go? Into, this is the inside of the cell, right? Does sodium want to go in or out of the cell? Sodium, does it want to move into the cell or out of the cell? It wants to move in, it wants to go down its concentration gradient. And potassium wants to move in or out of the cell? Out, wants to go down its concentration gradient. That would be normal, that would be entropy. But we are anti-entropy machines as living things, and we need to, for various reasons, and I'm going to show you one of them in a minute, we need to be able to pump sodium out and potassium in. And so what this does is, this particular protein provides ultimately a pathway, but that pathway is going to require energy, because I am going to have to go against concentration gradient. And so we take these, we'll hold up to 3, well not have to, must have 3. Because everything at the molecular level in biology comes down to what 2 words? Charge and shape. These things have a positive charge. They have a certain shape. When I load this with 3 of these, the effect of the charge on this protein causes it to change shape slightly. And so simply by adding 3 of these, it causes this protein to change shape, and now it can fit what we call ATP. So I'm going to draw some ATP here. I'm going to do 3 phosphates, 3 phosphates, and then we'll do like a big old adenosine. Okay. So that's ATP, adenosine triphosphate. This is my energy source. And again, we're going to learn more about ATP here in a moment. Okay, I'm going to show you a video. Actually, I guess I could do it right now. Do you remember in DNA and RNA, we had this little chant we did? Phosphate, sugar, nitrogen base, right? So let me draw this for RNA, okay? So my phosphate is, I think adenine is double ring. Okay, so we get a phosphate, and that bonds to ribose, which then bonds to adenosine, which I believe, it's been a while since I've drawn these. This is adenosine. So this is no particular charge, this is no charge, but phosphate has a negative charge. This is what makes it, these are the monomers that our RNA is made up of. But if I can start adding phosphates to this bad boy, it means I'm going to start putting negative charges next to each other. It's like putting two south poles of a magnet next to each other. They don't want to be next to each other. To hold them there, I have to have a lot of energy, right? Remember that we said, where's the energy at in all the food and stuff you eat? It's in the bonds. So I can put energy into this molecule by forcing it to bond together. It doesn't want to bond together. It doesn't want to stay there. These things want to go flying apart because they put negative charges next to each other. Well, it was bad enough when I put two negative charges next to each other. But what about when I, think about this. If you put two magnets of whatever, the same pole, it takes a certain amount of energy. But now if you're going to do three, that's even more, right? That's a lot more energy. So when I do adenosine triphosphate, wow, it took a lot more energy to put on this third one. Well, now if I can release this energy here, it's going to create movement, right? What happens when I let go of those south poles that are together? What do the magnets do? They go, right? The potential energy in the bond is turned into kinetic energy. Things literally will move, and so that's what's happening here. That kinetic energy, that potential energy is put into kinetic energy, and this thing moves, and literally this changes shape, right? It goes from this conformation. I'm going to draw one more of these. And it'll go to this conformation. I realize I made a mess of that, but you just got to go with me because I'm doing my best. Here we go. That's better. All right. Okay. So now, because of the change in shape, adding this and the kinetic energy forced and the charge forced the shape of this thing to change. Well, now when it did that, it changed the shape here, and it doesn't have such an affinity for these sodium ions anymore, and so they are going to get released. And I literally just moved. Because of the energy of ATP, I literally just pumped 3 of these to the outside. And now it's exposed here. If it were just exposed and that was all there was, nothing would ever happen. But it's not just that it's exposed, it's that these sites here have a high degree of affinity. They have the right charge and shape to fit potassium ions. And so 2 potassium ions will come and they'll fit right here. And when they do, that changes, that impacts the charge and shape of the protein, and it will snap back to its original shape, its original conformation, and it'll pump these 2 down here. Does that make sense? Do you believe me now when I said it's all about charge and shape? I mean, there's just no getting away from those 2 words. It's everything at the molecular level. So, to recap. I've got this pump. A pump requires energy, right? Energy source is going to be ATP. The energy in ATP is stored in the bonds, right? These negative charges don't like being near each other. When I release one, I'm going to transfer that energy. But I'm not even available to ATP until I have 3 of these sodium ions that are attached in the right spots. And that affects my conformation, the overall charge and shape of the protein, the parts move. Now I have a site that opens up, has a high affinity for ATP. The ATP comes, it connects, boom, hydrolysis of ATP. This thing snaps, goes to this conformation, this shape, releases my sodium ions, and now I have 2 spots, receptor sites, ligand receptor regions for potassium, which can attach there. When they do, their charge and shape affects the charge and shape of the protein, and the protein will snap back to its original conformation, no ATP there, and release them on the inside. And so I'm able to pump sodium out and potassium in. And it's a 3-to-2 ratio, right? I pump 3 out for 2 in. That even matters because I'm going to start to accumulate a lot more positive charge out here than I have in here. And this is how your neurons work. This is how you think. This is how like a bug gets on you and you feel it, you slap it. It's because this is happening. because you're able to pump these and create a chemical gradient, and your body uses that. I'm going to show you that in just a second. I do think we ought to look at that. Let's look at this video one more time. And now that we've kind of talked through all this, I think it will help. Also, your book just outlines the steps. You've got to know this. This sodium-potassium pump is everywhere in your body. Okay, let's take a look at this again.
The sodium-potassium pump works constantly to maintain a low concentration of sodium ions and a high concentration of potassium ions in the cytosol. This is achieved through the following steps. The sodium-potassium pump binds 3 sodium ions from within the cytosol. ATP then binds to the ATPase region of the pump and is hydrolyzed to ADP and a phosphate. This phosphate remains bound to the pump, phosphorylating it. Phosphorylation causes the pump to change in shape. The sodium ions are released into the extracellular fluid and 2 potassium ions bind to the pump. The phosphate group is then freed from the pump, causing the pump to return to its original shape and release the potassium ions into the cytosol. The pump thereby returns to its initial state, ready to repeat the process.
Okay, I want to show you 2 more videos, okay? I'll show you how ATP works.
Almost everything an organism does requires energy. But where does this energy come from? To answer this question, let's zoom in on one of this hummingbird's cells. Notice that each cell contains many mitochondria, which are often referred to as powerhouses of the cell. This is because mitochondria are responsible for producing most of the cell's energy in the form of ATP. ATP, or adenosine triphosphate, is really important because it serves as an energy carrier in cells. It's responsible for moving the energy found in chemical bonds of our foods to the areas of the cell that need energy. Without a constant supply of ATP, cells would quickly die. Let's zoom in to take a closer look at ATP's components. ATP is composed of a nitrogenous base called adenine, bound to the sugar ribose, and 3 phosphate groups. The bonds between the phosphate groups are high energy. An enormous amount of energy is packed into these bonds. When energy is needed, the terminal phosphate group is removed in a process called hydrolysis, releasing energy and forming adenosine diphosphate. The energy released from this reaction is used by the cell to carry out all the functions necessary to sustain life. Later, at the mitochondrial level, ADP and phosphate are recombined back into ATP, recharging the energy pathways of the cell. This cycle, from ATP to ADP and back again to ATP, is called the ATP cycle.
Okay? Let's go look at this.
Neurons communicate with each other through their dendrites and axon. Generally, incoming signals are received at dendrites, while outgoing signals travel along the axon to the nerve terminal. In order to achieve rapid communication over its long axon, the neuron sends electrical signals from the cell's body to the nerve terminal along the axon. These are known as nerve impulses, or action potentials. An action potential is essentially a brief reversal of electric polarity across the cell membrane.
Let me redraw that the way that it is in a way we understand here, okay? Okay, so they're drawing this long axon here. And what happens here is because you've got these sodium-potassium pumps, you're constantly pumping sodium out here. And this thing is just has tons and tons of sodium out here. Remember, it pumps 2 sodiums out for every 2 potassium in, right? So that creates an electrochemical gradient. Just simply got a lot more positive charges outside the cell than you do inside the cell. So it creates a net positive charge outside and a net negative charge inside. I wish there were a faster way for me to draw this. Sorry. Then we have lots, though less, of potassiums in here. Well, how did I get this difference? How did I create this concentration gradient that I have? Did it with the sodium-potassium pump. I actively transported sodium out, and I actively transported potassium in. I created this concentration gradient. And now your body is able to exploit this. This is literally how a nerve works. Because if I just open what we call a gated channel, if I just open the gate so that sodium can flood in, what's it going to do? Is it going to go in? Say yes. Yes. And if I open the gate so that potassium can go out, it's going to go out. And so all I have to do is just for a moment go and open it, and they flood in. Well, that flooding in creates an electrical impulse. A signal moves down this like that. And so you can you've got certain nerve, some of these that are huge, extending throughout your body. And all I have to do is have the right trigger. And all of a sudden this reacts, and that information flows down very, very rapidly. And then when it gets to the end, then you've got to do other things to transmit the signal. But at a molecular level, I got potassium in, I got sodium out. That happened because of the sodium-potassium pump. When I open these channels, they reverse, they flow until they're isotonic, right? Well, then that would mean I can't ever do anything with that nerve again unless I pump them back the way I need them to, right? I use the sodium-potassium pump. And so that's what happened. It's going to show you this on this, I do believe I've got the right video, that once a nerve fires, you've got to reset it basically, right? It's like, if I had a bow, an arrow, and I pulled it back, and I release, great, I shot an arrow, but to shoot the next arrow, I've got to reset the bow, right? And that's how this works. So let's, I'm going to back this up just slightly.
Electrical signal from the cell's body to the nerve terminal along the axon. These are known as nerve impulses or action potentials. An action potential is essentially a brief reversal of electric polarity across the cell membrane. Cells are polarized, meaning there is an electrical voltage across the cell membrane. In a resting neuron, the typical voltage, known as the resting membrane potential, is about negative 70 millivolts.
You don't even know that.
The negative value means the cell is more negative on the inside. At this resting state, there are concentration gradients of sodium and potassium across the cell membrane. More sodium outside the cell and more potassium inside the cell. These gradients are maintained by the sodium-potassium pump, which constantly brings potassium in and pumps sodium out of the cell. A neuron is typically stimulated at dendrites, and the signals spread through the soma. Excitatory signals at dendrites open ligand-gated sodium channels and allow sodium to flow into the cell. This neutralizes some of the negative charge inside the cell and makes the membrane voltage less negative. This is known as depolarization, as the cell membrane becomes less polarized. The influx of sodium diffuses inside the neuron and produces a current that travels towards the axon hillock. If the summation of all input signals is excitatory and is strong enough when it reaches the axon hillock, an action potential is generated and travels down the axon to the nerve terminal. The axon hillock is also known as the cell's trigger zone, as this is where action potentials usually start. This is because action potentials are produced by voltage-gated ion channels that are most concentrated at the axon hillock. Voltage-gated ion channels are passageways for ions in and out of the cell, and as their name suggests, are regulated by membrane voltage. They open at some values of the membrane potential and close at others. For an action potential to be generated, the signal must be strong enough to bring the membrane voltage to a...
It's probably more than you guys need already. All right. My purpose in showing you that video is to show you, I mean, it's literally how muscles contract and how brains think, is you've got to create this difference in concentration gradient, active transport. And then it opens a channel and allows it to just move back down. That's passive transport, right? Or facilitated diffusion, right? So this is not diffusion, this is active transport. But those other ones that it showed you at the end, that's just diffusion. It's just opening it and letting it pass. And when it does that, it creates this electrical current, and that's how thinking happens, that's how muscles get triggered and fired. If you can't do this, you can't do that. If you can't do this, you can't think, you can't move, you can't eat, you can't survive. If you had some genetic problem to where your sodium potassium pumps weren't made properly, they didn't have the right charge and shape, well, you would never be born. It's way out ahead of where we're at, but I mean, you guys are familiar with the concept of a miscarriage, right? You know, so an egg is fertilized by a sperm, but at whatever stage that baby can't survive anymore. What's usually happening there is there's some kind of a protein that's not being made correctly. So there's a change to the DNA, which ultimately leads to a change in the protein, and there's often some very particular protein that it must have that's just not functioning right, and so it can't do homeostasis. Oh, it's not written up there anymore, but it can't do homeostasis, and so it can't make it. Okay. Most genetic diseases that we talk about, that's really what we mean. There's some change to the DNA, which leads to some change to the protein at the charge and shape level, so it can't do what is necessary to maintain homeostasis. All right. We talked about phagocytosis and pinocytosis, right? We did that actually way back even in cells, and we reviewed it. These are Greek words. Phago means to eat, pino means to drink. So cellular eating is phagocytosis, cellular drinking is pinocytosis. You got to know those terms. Really, that is it for this unit over organelles and over membranes. Do you all have any questions? I'll set you free. That'll be our class.